Today we're going to talk about sexual reproduction. If you can understand this standard, it's going to help you a lot on your state test. It'll help you identify questions and understand them a lot better. So this video should help you a ton. Alright, so we're going to start off by looking at some vocabulary words. If I were you, I would pause this video and write down these words. It's going to help you a lot. So our first word is gamete. This is sex cells. In females, you have your eggs. So an egg would be a gamete. It's a sex cell for women. And sperm is a sex cell for males. Okay, so sperm would be your male gamete. All right, meiosis is actually the process of sexual reproduction. So we're going to break that down today. That's really what this video is going to be about. You might often hear this called meiosis, but I like to call it meiosis just so I don't get it mixed up with mitosis. Okay, so meiosis is what we're going to call in this video. All right, it produces 4 haploid cells. Now, what are haploid cells? Haploid is represented by a little n. This is also, I've seen it on some practice questions for the state test. So haploid, every time you see it, represented by a little n, means that it's one set of chromosomes. So it contains half the number of original chromosomes. Okay, diploid, I try to think of die like a set of dice, 2 sets of chromosomes. That is represented by a 2n. This set contains chromosomes from each parent. So in your diploid sets of chromosomes, you have chromosomes from mom and chromosomes from dad. Okay. Our last word is going to be crossing over. This is a process where homologous chromosomes trade information. So homologous chromosomes are, think of homo means same. So homologous chromosomes are like chromosomes from your mom and dad that kind of have some of the same information. Okay, so let's go ahead and look on the board. We're going to go through each of the phases of meiosis. So this is how you were formed, how baby dogs are formed, okay? So what happens is a mom's egg and a dad's sperm meet together and they make this little egg, okay? So that's where you start. So prophase 1 is the beginning of meiosis. We have meiosis 1 and meiosis 2. Always start with the smaller number. So we'll start with 1. All right, prophase 1 is where you have your mom and dad homologous. Homo means same. Some of their same chromosomes get to swap some information. So I don't know if you can see those little pink and blue X's, but the blue is going to represent the dad chromosomes and pink for mom chromosomes. All right, so what happens in crossing over is these same chromosomes or similar information on these chromosomes start switching their information. So you'll see me change this blue to a little bit of the pink at the top and this a little bit of the blue. So mom and dad are swapping information. That is crossing over. This can only happen in prophase 1. All right, we can do it on more than one too. I'll go ahead, switch some more. This creates more of a genetic diversity, okay? So after that, we can see that our nuclear membrane is starting to disappear in prophase 1, and our centrioles right here really don't have anything that they're doing yet. All right, next we're going to go to metaphase, okay? Metaphase, you have no nuclear membrane. Your chromosomes are starting to line up in the mama middle. I like to think of mama middle, mama metaphase, okay? So your chromosomes are lining up in the middle. Okay, we have 2 rows in the very middle. All right, don't forget that you have already swapped some information. So to save time, I'm just gonna blot some on there. All right, in anaphase, they are trying to move away from each other. So A4, away. These chromosomes are gonna pull away from each other with these little things called spindle fibers. They're gonna reach out like those little games that you play in the arcade where it grabs the little toy. They're going to reach out and they're going to grab these chromosomes and pull them away. All right. In telophase, this is your last phase. Telophase 1, what happens is these chromosomes are going to start coming into different cells. So the chromosomes right here are going to pull to one cell. The chromosomes here are going to pull to another cell. So that's what's happening right here. You have your nuclear membrane starting to form again. You have your new genetically diverse chromosomes separating. Remember, this is where we crossed over earlier. All right, and then this little thing called cytokinesis is going to start occurring, and it's going to actually pull these cells apart. So cytokinesis is really the process of them making two individual new cells. So I'm just going to be a little quicker here. All right, and now we have two cells. But that's not how you end meiosis. You have to go through a whole nother process of meiosis called meiosis 2. Okay, in meiosis 2, you're going to start out with prophase again. Prophase, remember, you cannot have crossing over in any other phase but prophase 1. So we do not cross over anymore on prophase 2. Our chromosomes will remain just like they were. Wrong color. Our chromosomes will remain just like they were over here in cytokinesis from meiosis 1. All right, your nuclear membrane again is the same as prophase 1. It's starting to disappear in prophase 2. Your centrioles are just sitting there, not really doing anything yet. Then we move to metaphase 2. This is the middle phase where your chromosomes are going to line up in the middle. All right, there's a little difference here. Then over here, we have two rows. These are when mom and dad's chromosomes weren't separated yet. Now we've got them kind of separating over here in the metaphase 2. All right, in anaphase 2, these are going to pull away from each other. So here, your sister chromosomes pulled apart from each other. Here, your sister chromosomes are going to pull apart, but we're not going to pull a chromosome here and a chromosome there and a chromosome here. We're going to pull them apart and make them chromatids. That's the singular version of chromosome. Kind of did them in a little different order, but it's okay. Okay, and then over here, your chromosomes are going to turn to chromatids. Can you see over here? I just want to show you, this is a chromosome, and if you change a chromosome, if you split it in half, it turns into a chromatid and a chromatid. Okay, so when I say chromatid, I'm talking about half of a chromosome. All right, now back to here. We're going to go to telophase 2, last phase. In telophase 2, our chromosomes are going to start combining into one cell here, one cell here, one cell here, one cell here. So you're going to end up with 4 cells. Let's go ahead and do it like this. Just trying to get a little quicker. All right, so now your chromatids, are pulled apart into different cells. And now we're going to close up this nuclear membrane in cytokinesis, and we're going to have 4 new cells. We have 4 new nuclei, and inside these cells are going to be your chromatids, and they have changed from a diploid number that we started off with to a haploid number. Now this means that you have half the original number of chromosomes and you have half the information. So the information is split up from your mom and dad chromosomes and it's totally just intertwined, and you have unique new cells.